Hey, thanks for coming out. The sort of the target time frame for these usually is about an hour and a half, and that includes whatever questions you might have. So if you have to leave before that, I'll certainly understand. But you know, sometimes they go even 2 hours, but an hour and a half is typically enough. So, and I do like to keep it informal. The handout that you've been provided is has a number of things that you might find interesting. There's a lot in there actually, and we can send it to you as a PDF. The county agents can send as a PDF if you want to click on some of these links, or you can just find, like for example, 20 points of broad scientific consensus on GE crops. You can find that on the web yourself and follow the links yourself. All right, I think that's. Oh, and I guess I will say. The GMOs stands for what? What does that stand for? Genetically modified organism, right? And that's a very commonly used phrase and term. Most scientists prefer to talk about genetic engineering. It kind of means the same thing to the public, but we prefer to talk about genetic engineering, even if it's understood as GMOs, because all organisms are genetically modified. There's nothing wrong with genetic modification. Anyway, so we want to distinguish normal genetic modification from the genetic engineering, which is where humans are directing the changes that happen. So one of the most important things I start with is a list of all the funding that I've received to do work and to talk about GMOs, or genetically engineered crops, which is nothing. So yeah, so I'm in fact I've turned down. Well, let me show you. This organization represents itself as a nonprofit organization. I trust that they are. They contacted me and said, "Would you write an article on industry relations and research?" And I said, "Sure, I'd be glad to." And they said, "We'll pay you a $1,000 honorarium." And I said, "No. I don't want the money. I want to be able to continue to say that I'm independent of any conflicts of interest in this topic." And I said, "But you can donate it to Human Rights Watch." And they said, "No, that's illegal. We have to give it to you." So I said, "Okay," and I received their money, and I turned it right around to Human Rights Watch. So at this point, I've given up, foregone $2,000 in funds from nonprofit organizations, like education organizations. And there's like immense distance between me and Monsanto. Okay, if you want to read more, it's all online. My relationships with industry over the years. I've had industry funding, not on GMOs, on other topics. I'm very proud of the work that I did, always on behalf of the public. But you can read all about it. It's online. Anybody in the world can read about it. So when you hear me say something, you're getting my judgment as a public scientist. And my responsibility is to you, and that's what I do. And I'm actually. It sounds immodest to say, but I'm a good scientist too. And so my work is peer reviewed, and everything I say is very defensible in the scientific literature. One of the things that I think is important to understand too is that genetic engineering is actually a big part of our lives already. And so if you know a diabetic, it is very likely that they're taking insulin that was produced in a genetically engineered bacterium. And frankly, I would prefer that if I were a diabetic, because then you're getting a more pure product than insulin extracted from pancreases from livestock. So that's one well-known and long-term example. If you eat hard cheeses, most of the enzyme that is used to create hard cheeses is from a genetically engineered organism, yeast. Yeah, it's a yeast. Okay. So again, it's another part of our lives. And therapeutics, vaccines, medications often involve some form of genetic engineering, and more of that will happen because it gives us control over what we're trying to do and what we're trying to generate. So this is this is really an important development. Okay, so I'm kind of in this introductory part bouncing around on different topics and saying just a little bit about each one. The question came up about Roundup Ready crops, right? So let me explain. Who's heard of Roundup Ready crops, first of all? Okay, about half, roughly half. So the most common use of genetic engineering in crops is right here, Roundup Ready. Roundup is an herbicide that is very effective. So it's killed all these weeds, okay, except for the corn, which is not a weed, of course. And the active ingredient in Roundup is called glyphosate. So it's good to recognize the name in the context of this talk, because I may say glyphosate tolerance instead of Roundup tolerance. So what Monsanto did, so this is one of the ways they made a lot of money using these technologies, is they took a bacterium that is naturally occurring in the soil, totally natural bacterium, and they took one gene out of that bacterium, and that gene they put it in corn, and that gene helps the plant, the corn, tolerate the application of glyphosate. So instead of dying, it's able to continue to live by having that one enzyme from this bacterium. So for growers, this has been, I don't know if I'm overstating it to say it's been sort of almost a magical technology. You could spray the field full of weeds, and you would get complete control, nearly complete control. And this technology has been very popular worldwide, very popular. Brazilian farmers, when they couldn't use it, but Argentinian farmers could, they went to Argentina in their trucks and they bought seed and they brought it back illegally. That's how popular this is. So I want you to understand, if you have any question as to why growers would do this, talk to a Western Kentucky grain farmer. You got lots of them, okay? Because it was so successful, glyphosate, the active ingredient, has been widely used. And not surprisingly, growers really like the technology. It provides simple weed control, it's inexpensive, and it's effective. And it offers other benefits. So I'm giving you the pros here. And so some of those benefits include better erosion control, because you don't have to always plow the field, or till the field, I should say. So better erosion control. Actually, if you go back to the mid-1990s, the herbicide programs being used were more impactful, negatively impactful on the environment. So, at the time, people thought of Roundup Ready crops, Roundup tolerant crops, as really being a good idea. So the two downsides that I think are important to mention, this is sort of the other side of the coin now. One is this, is it risky to consume glyphosate? Because when you apply glyphosate to those young corn plants, some trace amount is going to end up in the grain, as is true for any pesticide. Some trace amount might end up in the grain. So, so the question is it risky? A lot of studies have been done on Roundup tolerance, a lot of times published not in agricultural journals, but journals that are associated with the medical profession or the toxicology profession. So by and large, I think there's a lot of evidence showing that glyphosate is safe to eat at the levels we do. The World Health, a branch of the World Health Organization in 2015 declared from their analysis that glyphosate was a probable human carcinogen. By and large, most regulatory agencies that have done their own analyses in response to that analysis by the World Health Organization, most, like the EPA, the European Food Safety Administration, and so on, most have concluded, no, it's not a carcinogen. But that one did. And California recently, the state of California recently declared it sort of a me too. Oh, if the World Health Organization, that particular branch, says it's a carcinogen, so do we. So, you could easily convince yourself that it's uncertain, right? There's some uncertainty in terms of the safety of glyphosate. And I won't try to talk you out of that. I think there is a little bit of uncertainty. Personally, I'm not too worried about it at this stage, but am attentive to what the scientists are continuing to do. All right, so that's one downside of this Roundup Ready crop. And I will say, if ever glyphosate is found to be more harmful than we thought, then the problem is not the genetic engineering. The problem is the glyphosate. Okay, that's a really important distinction. So what happens, here's the other downside to Roundup Ready crops. What happens if you spray a toxin or apply a toxin over and over to the target population? It becomes resistant. If it doesn't, it dies out. So yeah, it has this strong selective pressure to become resistant. And that's what's happened with Roundup Ready crops. Even though it was not expected to happen, it has. So weeds have, this is mare's tail, growing in soybeans, just overgrowing the soybeans, and it's been sprayed with Roundup. And now this has been sprayed with another herbicide program. So growers can do other things to control these weeds. It's not, that all is lost, but now we've got glyphosate tolerance widely occurring throughout Kentucky and elsewhere. So now what's happening is crops are being engineered to be tolerant to Roundup and maybe 2,4-D, another active ingredient. So in 10 years, 15 years, we might have resistance to those combinations, right? And then maybe in 10 years there'll be a third. So I think this is a downside. It's a pesticide treadmill. It's not a sustainable way to control weeds. I don't mean that to say we shouldn't do it. Because again, the Western Kentucky farmers have reasons for doing it, and they will tell you what they are. But it isn't a sustainable solution. We're not going to be doing this in 50 years. I don't really have the easy answers for weeds. Well, I have some ideas. But this, if you ask me, give me an example of a GMO crop or a genetically engineered crop that is beneficial, I would not point to this. Because it's too mixed. There's too much pros and cons, even though both sides are right. All right, so bear with me while I get my, where to go? All right, I'll use this one. All right, so this is a non-GMO apple. Okay, just purchased out of Kroger's and just a normal apple. So,
how do you know that?
Well, I bought it. So there are no GMO apples. Whoever asked that question. Okay. There are no genetically engineered apples on the marketplace. There may have been some in a research mode. I don't recall ever. Well, no, Arctic apple. I'll talk about Arctic apple during the Q&A. This one is not. There's no, is this Honeycrisp? I can't remember the brand. Anyway, so raise your hand if you think I'm eating DNA. Just one?
What'd you say?
Raise your hand if you think I'm eating DNA. Okay, I see some hands going up. Good. All right. So what kind of DNA am I eating? Apple DNA. And probably bacterial DNA, because there's bacteria on the surface of this. Plus my hands have probably some other species of bacteria. And maybe some yeast on the surface, so I'm eating probably yeast DNA. And maybe some insect eggs, so I'm eating insect DNA. So, oh yeah. Yeah. I actually did, but still there could be some stuck, right? So I think it's really important to start with this point, that DNA is in our food. It's a very natural part of our food. And DNA, or its sister molecule, RNA, are completely safe to eat. They're completely safe to eat. So now, I'm not saying that DNA changes can't cause harm in the plant. I'm saying the DNA itself is not toxic. We can eat lots and lots of DNA and never have any harm. So is that okay? Everybody get that? Okay. This slide is to illustrate a point that really surprised me, and that if we think we are doing crazy things with DNA, wait till you see what nature does. And this is just one example, and much of it is very recent kinds of findings, but not all. Some goes back a number of years. So who likes sweet potatoes? Okay, most of us, yeah. So what we've discovered, or what scientists have discovered, is that sweet potatoes all the cultivated sweet potatoes that have been tested worldwide have at least 4 genes from a bacterium, at least 4 genes in their genetics. So sweet potato is actually a natural GMO. And that probably surprises some of you, maybe all of you. It did me. That's why I got into this. I actually started reading things about these kinds of genetic wacky things going on out in the, beyond our laboratories. And I said, man, this is really bizarre. And I wonder if the public knows this. So, and that's when I proposed to the college administration, would you like me to work on outreach on this topic? And they said, yes, it's urgent. So that's how that all started. So I could give you a million examples, but I'll stop with that. Okay, here's another introductory point that is really important, and that is the way that biologists see DNA is not as a magic substance and not as a mysterious substance, but as chemical information. That's the way biologists see DNA. So, so there are 26 letters in the written alphabet. How many letters are there in the DNA alphabet? Somebody knows. No?
4.
4, right, exactly. So everything with DNA comes down to the arrangement of 4 letters, okay? Pretty amazing. The best way to explain how, again, I think I'm representing the scientific community in this. The best way to, in fact, I've had this vetted by scientists, so I feel very confident in it. The best way to understand genetic engineering, it's like word processing. So you probably all have the experience of knowing that you could copy a sentence. So the sentence is the gene, okay? Not the individual letters, but the sentence is the gene. So you could take a sentence and copy it from a file and put it into another file, computer file, completely different. And it may come from a history book and go into a cookbook. And that may seem really bizarre, but that's what we do. And so where the sentence goes really might matter. Suppose it ends up in the introduction, and you read the book and you say, it reads better. Or suppose that sentence you pasted into a recipe, and you've just ruined the recipe, so it's not going to perform well. The same happens with genetics. It isn't the transfer of the information that matters. It's what it does in its new place that matters. Okay. So, and again, stop if something I say seems puzzling or nonsensical. Okay, so, right. Okay, so let me explain what I'm doing here. So what I have is, and this is also one of the really most important points. I really pruned out a lot of information into this talk. But all right, so this deck of cards is a crop that grows in Kentucky, whatever crop you want it to be, and every card is a gene. Okay, so they're all the red, genes that represent this plant, and we really like this plant. It performs well in Kentucky. It yields well, but it doesn't have resistance to a particular disease. We're worried about that. So what we do then is we have this plant, which is a wild relative. It's a different species, but it will hybridize with that one. And it's not going to perform well in Kentucky. It's not going to taste good. It's not going to survive our winters. But it has the 5 of diamonds. And the 5 of diamonds is a resistance gene for the disease we're concerned about. So it's going to be in there somewhere, right? And so what we'll do, using traditional breeding, is we will hybridize the two plants. And you can see we've clearly mixed up the genetics of the plants immensely. And that's okay. That's good. That's what happens in breeding. But we've really made a kind of a potential mess if you want to look at the red plant as the pure plant. And so we can backcross. I won't explain backcrossing unless you have the interest later. But we can get rid of most of the blue genetics through backcrossing. We can't get rid of it all. We've permanently altered the red parent. So maybe that's not a bad thing because we can look at the, the plants that result and try to find one that is useful. So let me find my 5 of diamonds. I need my 5 of diamonds. So we're going to do this a different way. Should have done this before. All right, there it is, 5 of diamonds. All right, so here we have the red parent as before. So it's the red parent. And here we have the blue parent as before. And there's the 5 of diamonds. So instead of hybridizing, why don't I just copy this in the laboratory and insert it into the red parent? And that's what genetic engineering is. It's nothing more than that. So, so what I was trying to illustrate is, and this is a finding that is well established, and that is genetic engineering is actually more precise than conventional breeding. Yeah, it's actually more precise. Or causes less, let's say, yeah, it is more precise actually, but it also causes less impact on the plant. And that surprised me, as it may surprise you. But if you think about it for very long, you realize it's not so surprising. Because, look at what we did with hybridization. I mean, this is a sort of a crude way of thinking about hybridization, but it gets the point across. So we've done a lot of genetic change. And here's what we've done with genetic engineering. We've just put in 1 gene. So, so actually, kind of makes sense after all.
So,
Yeah.
Paul, would it be safe to say that some of the produce that you would buy at Kroger or at Walmart comes from hybrid crops?
Yes.
Yes.
All.
So those products that are just labeled normal produce products
Yeah. Mm-hmm.
would be from hybridization.
Well, somewhere along the way, they often do derive from a hybridization event. There are actually, if we looked carefully at, and I've done this, if you look carefully at breeding program, conventional breeding, okay, so here's genetic engineering, and here's the population of techniques that we're going to say are conventional breeding. Everything other than genetic engineering falls into this pool of conventional breeding. Conventional breeding can be as ancient as going out and selecting the plants that are growing taller, selecting seeds from the plants that are growing taller. And then the genetic modifications are happening naturally. And you don't know what they are, but you just know the plant grew taller. Some of these conventional breeding techniques are crazy. Not hybridization seems tame. Mutation breeding. So you take seeds of the plant you're interested in, and you expose it to gamma irradiation, or soak it in chemical mutation-causing chemicals. And the seeds that survive, you grow those up, and you select out the ones that you think might be good parents for further breeding. I mean, that's just removing embryos from plants that would die because they shouldn't be crossing, but we are forcing them to cross. Smashing together plant cells in the laboratory. All of those are conventional breeding techniques. And all of those are okay because they're non-GMO. So the point is that they're all good techniques. They're all good techniques. But we're doing more disruption to the plant. And we have less clue as to what we're doing when we do conventional breeding. We often do not know what changes we're making. We just like the way the plant looks. And that's all okay.
You don't hear the negativity
Right.
about the conventional breeding, even though it can be more disruptive.
Right.
Like you hear about genetic
Right.
engineering.
Right. Which is why I bring up this whole point. Yeah.
Now, I'm assuming you're talking about nutrition.
No, I mean, you can get, so it will affect nutrition, but you can get more disruption through conventional breeding to the genetics of the plant, to the RNA species which follow from the genes, the proteins from the plant that the plant produces, and the metabolites that it produces. Every aspect of its growth is potentially changeable. And it's more changeable through conventional breeding in terms of the disruption it causes. So that can lead to a more nutritious plant or a less nutritious plant. So can genetic engineering. But you're more likely to have those unexpected changes with conventional breeding. I'm not saying, now I might sound like a cheerleader for genetic engineering. There are risks, and we're going to talk about those, but these are the reasons why a lot of scientists say, you know, we're not really doing a fair evaluation of you know, how to best use these techniques.
Well, there was a show on TV, and by the time you got done with it, you walked away feeling like you should never eat corn again.
Because it was genetically engineered? Yeah. Well, the only, I'd say, you know, even if we want to be extremely careful, you might avoid corn products because of the trace amounts of glyphosate. You could. But genetic engineering, I'll show you some more on the safety issue. Another general concept that I think is important to understand, you know, in an audience like this, you've come expecting some depth, is the concept of a gene pool. So a gene pool is the pool of organisms that breed freely. So dogs are a gene pool, right? So I want you to understand that, because this is, you can actually use genes from different sources, because remember, it's copying and pasting, okay? So, so if you take an apple gene and put it into another apple by genetic engineering, that's called the cis gene. That's from the breeding pool. And I think it's well understood I know it's well understood among scientists that a cis gene could move of its own through natural processes or through conventional breeding. So if you're working genetically engineering with cis genes, that's a totally different, much, you know, you might not be able to distinguish them from natural processes. Transgenes are genes that come from outside the breeding pool. They've been transferred, transgenes. And so remember I said that the Roundup Ready trait came from a bacterium? Well, that would be a case of a transgene, a really good case of a transgene, because a bacterium is not going to mate with an apple. Now, if you can also take a gene from corn and put it into apple, and that is a transgene too, because corn does not mate with apple. But, you know, typically surveys show that people are less sort of creeped out by genes from plants that we already eat, even though a gene from a bacterium to a biologist is just chemical information. And, you know, if it does a good thing for us, biologists typically are like, you know, good with it. Okay, so how do we do plant transformation? How do we engineer plants? I'm going to show you 3 ways, and the first 2 are right here. These are the older techniques. They're, you know, maybe 25, 30 years old, but they're still useful. So one is this one, and this one this is a root, and those are bacterial cells, and when they're standing on the end, it's a natural bacterium, widely occurring in Kentucky and elsewhere. When they're standing on their end, they're inserting their own DNA into the plant. And I know, it is a wow moment. That's what scientists said. When they studied this, I think it was like in the 1960s maybe, they realized what was happening, or maybe the early 70s. They realized that the plant was being, this is where we got the idea of genetic engineering. Somebody said, hey, if the bacterium inserts its own DNA, why don't we take that DNA chunk out and put in another gene? And so we learned genetic engineering from this bacterium. And it works well, it still works well. The only downside to this technique is you control the gene that you insert, and therefore what it should be doing, but you don't control where it goes. So you do maybe 100 or 500 plants and hope and expect, you know, some of those will be useful. The other older technique is the gene gun. It's kind of a violent way to do this, but you take micro pellets of tungsten and you coat them with DNA, the DNA you're interested in, and you shoot them into leaves. And then some of those cells survive that process, and then they grow up to be plants that have the gene of interest. So you can do it by either method. The only downside is you're inserting whole genes randomly into the organism, into the plant. Who has heard of CRISPR? Okay, this right here is the most important thing that you can take away, even though there's a lot I think is important. So there's a new way to manipulate genomes, genetics, it's called CRISPR, or genome editing, and it's really huge. The little display I'm going to show you is not going to impress you with the depth of the revolution that's taking place in biology. So here we have a misspelling. Wine is made from grape juice. Well, with the older techniques, the only thing you could do is insert a whole gene somewhere in the genome. And that may be useful or it may not. But here, with genome editing, we can go in and make a precise, targeted change. Right? Or we could put in a whole sentence if we wanted, but we could say this is where we want that sentence, right here. So it's a very targeted and precise way to make genetic changes. Now maybe the genetic change that needs to occur is to remove some genetics. So maybe this is a genetic defect, right, in humans. I mean, a lot of research is being done with genetic diseases in humans using CRISPR. And it's only like 5 years old or 4 years old. It's incredible what's happening. And so, anyway, so remove but we didn't remove DNA at random. We removed just one letter. So CRISPR technologies, genome editing, are really, really huge, whether we ever engineer another plant or not. So how would this be useful? So let's take what this is. Let's start with the top line. So there are the 4 letters of DNA in that line. And lots of T's and C's and G's and A's. Okay? Now that is a partial gene sequence from wheat. And this partial gene sequence is from a particular gene that makes the plant susceptible to powdery mildew. Okay? So they went after this gene knowing what it did. They didn't just pick it at random. And what they did was they used genome editing to create a couple additional plants that had mutations. Mutation happens all the time in nature, and we can cause mutation if we so choose. And they did. So you see this, the middle line is one of the plants that they genome edited, and it's missing 3 letters, just 3 letters. And that change represents, in the whole wheat genetics, a 0.0000001% change in the genetics. So extremely modest changes are as a result of this. And in this plant, they only deleted 1 letter. Okay? So what was the outcome of that? Because again, they selected this gene. They didn't just do it at random. And so the outcome was what they expected. Lots of powdery mildew where they didn't engineer, and with as little as 1 letter being deleted, they engineered a plant that was resistant to powdery mildew. So if like our wheat growers use pesticides, fungicides against powdery mildew, there you go. I mean, I like that better personally. And so will the growers. That's not commercialized yet. But that was a proof of concept. Okay, let's talk a bit. And again, just do interrupt me if you have questions. Okay. So anyway, this is about food safety. So the most clearest way I can say, to say that as I understand the literature, that genetic engineering is safe is to say that I just don't have any concerns about my family eating genetically engineered crops. I do have concerns if they eat too much sugar or don't eat enough fiber. They should eat lots of fruits and vegetables. All those things that nutritionists say. Those are get a good night's sleep. Drink plenty of water. But genetic engineering, yeah, I'll show you what the major societies say. So So this is a really effective communication. And it's scary. And when you touch on people's fear, that makes a big difference, right? And I want safe food. I want to tell you, scientists want safe food too. We're with you on that. Is genetic engineering, does it create unsafe food? Well, that's not what the science says, by and large, okay? And I read the papers that say there is a problem too. I look at those and see, were those, are they right? You know, have they done a good job in their experiments? So, this idea of frankenfoods, it's really effective, but it doesn't represent scientific findings. And I'm going to show you in just a minute some evidence for that, just a tiny bit of evidence. But if this surprises you, then let me ask you to remember that there's no harm to the computer in moving moving sentences around, and it's the same thing with genetic engineering. It does it. It's okay. I think I've said enough about that. And remember too that we cause less disruption with genetic engineering. We cause less disruption to the plant. So we're actually maybe more likely to have unexpected problems with conventional breeding. I don't know. It seems like a good argument.
I was just wondering, where did the misconception start
Yeah.
when it comes to the GMOs?
Yeah, that's a really good question. And I'll give you my sort of nutshell response, and that is, I think there are going to be dissertations written about this topic and time in the future. But I typically stay away from trying to speculate on what dynamics have happened over the decades. I have friends that think that they're dangerous, and they're, you know, I almost lost a dear friend because of it, but she's been able to, I think, recognize that you know, I've reached my position honestly and through careful investigation. So I just talk about the science. Yeah, and I'm giving you the peer-reviewed science, basically. There are outliers, just like any topic, you know, any topic you can find scientists. If you seek out somebody to validate a particular position, you can find it. But I'm representing the large body of scientists throughout the world.
Now that we're on this topic, could you address this, the research findings of this senior level group in MIT a couple of years ago that said that Roundup was responsible, when Roundup, when used in combination with vaccines, is responsible for this explosion of autism?
Yeah.
Could you address that?
Yeah. Yeah, it's not in my presentations. I think that was Senef is the last name of the researcher.
I'm really ignorant.
It is not. I've looked at those papers. I've looked at follow-up critiques by credentialed scientists, and I don't think there's any evidence that's held up at all. So it's a, you know, any concern, any topic is a valid topic to pursue, but I just find that falling apart. And I want to stress, I really do pursue the questions. So we have, to give you an example, I don't have slides on gene silencing. It's a technology that, well, it's a natural process, and we're just taking advantage of it as humans when we engineer plants to silence certain genes, right? When I read that this particular researcher from the University of South Carolina, very respected scientist, had concerns about gene silencing and safety, I have to tell you what I did. I found money to invite her to give a seminar, 2 seminars, to the public and to the room full of scientists. I read intensely. I took her out to dinner several times when she visited and quizzed her. And I did a series, over a period of years, I did a series of investigations of the body of literature. And she's an excellent virologist, but her work has not held up against evaluation by really top-notch RNA specialists. And there are papers from the anti-GMO literature that have been retracted. Yeah, so we got to take everything seriously, but sometimes there's a reason why major scientific societies have rejected claims like that.
Well, that information from the MIT group is still much cited.
Yes,
And I know
it
there's
is.
a professor emeritus at Purdue, plant pathologist, who subscribes to that.
Yes, Huber. Yeah, yeah. So all right. So I'm gonna respond.
You're
Yeah.
saying this lady's was more like on his side, where she was trying to prove that it was causing problems, or
Yeah, yeah.
and you're saying it's not been founded.
It's not held up in the world of peer-reviewed science. That's what I'm saying.
I mean, there's so many cancers. There's so many things going on. Every time
Right.
I put hairspray on my hair, I
Right.
wonder.
So
What am I breathing?
So don't we want to be sure what those causes are, right? And
Yeah.
so if it were genetic engineering, I would be the first to tell you. I wouldn't drive all this way and be home away from my family unless, I mean, I would tell you, I don't have any financial conflict of interest. Yeah, the Seneff work has, you look for her name, Seneff, S-E-N-E-F-F, and then look for the follow-ups. And remember, I have access to papers in the library system, the University of Kentucky. So I get to pursue it to any depth I want. So since you brought it up, I'm going to identify him. Don Huber had a Purdue emeritus, had an illustrious career as a plant pathologist. I don't understand what's going on with the claims he's making. For, I think it's 7 years, he's been claiming, making very unusual claims about genetic engineering. Well, I'm telling you, because I've interviewed with the New York Times on this issue and Discover magazine, and it always comes down to this. I say to the journalists, he has not presented that, to my knowledge, in a major scientific conference. He has not published this. So until we have something tangible, it's just his word. And he cites European scientists, but sorry, you got to publish it. Or you got to present it at the American Microbiology Society or something. So I'm a little frustrated.
The MIT group has published theirs. To my mind,
Right.
MIT is the creme de creme.
Yes,
of science.
it is. Doesn't mean that that particular paper or paper series has held up.
It's a group, one person.
Right. Seneff is the principal. I believe Seneff is the principal author, first or last. Yeah, I mean, you have to look for the follow-up, work, published work. So, yeah.
I wanted to touch on this gentleman's question about when did it start, kind of the fear of human, because I work with the Extension Office, and I do have a lot of clients and consumers that ask, well, I read this on Facebook, I saw
Right.
this on the internet. And I think a lot of that comes from genetic engineering being new to people. And so there are studies that have been done that may show some concern. And so people
Sure.
will cherry pick those studies and say, well,
Right.
this study says there's a problem. And then the media or social media will get a hold of that and run with it.
Right.
Because it thrives on people's fears about food not being safe.
Yeah.
Whereas, truthfully, that study that they cherry picked that said there were concerns has not held up. The
Right.
scientific findings have not shown
Yeah.
that genetic engineering is harmful.
Right, and that's the part that that you wouldn't even know about, and even then it might be hard for you to access, unless you go through the University of, maybe the UK would provide you access, but
But don't you have to admit that we really don't know, except maybe 50 years from now?
well, I guess, I mean, we don't know, but I mean, we've done. So let me show you this. Let me show you a few slides, and then I'll try to get that. So I mean, we never know. Truly, we never know if cell phones aren't going to cause, right, cancer, but we see benefit to them, and it may take 50 years to know. So. So part of what I want to say in response is this, and that is this to me has risen to the height of a principle of biology, and that is because I see it and I've seen it for decades from excellent scientists. Genetic change, what matters is not how the genetic change was made, but what the genetic change does, just like the copying the sentence into another file. That's really important. So I guarantee you we could create a plant that's toxic through genetic engineering. we can also create a plant that's toxic through conventional breeding. And it's been done. Accidentally, but it's been done. So what matters is not the way you make the genetic change, but what the genetic change does. So I want to give you just a taste of, I'm not even going to show you studies. I'm going to show you reviews, just a couple reviews, and they're in this document that I've given you. You can link to those. So this is a 600-page review by the National Academy of Sciences. I have a lot of respect for the people that are asked to serve on these reviews by the National Academy of Sciences. And they're like me. They're going to tell you if there's a problem. But they're saying that there's no substantiated evidence, right, that there's one form of changing the genetics is different from the other. And that's the U.S. Academy of Sciences. But in Europe, they don't, what do they say? Because the people don't like genetic engineering. So if you go to the equivalent scientific body, they're saying the same thing. And you can read it for yourself and tell me if I've misrepresented what they say. Now that's puzzling. Wait a minute now. I thought that France and Italy and Germany, are very anti-GMO, and they are. But their scientists see the same published literature that we do. And that's what they say. That's what their scientific body says. I'm going to let that. So yeah, I mean, ultimately, we don't know. We never know whether anything. I mean, I would say, to play devil's advocate, do we really know that conventional breeding is safe? Because, yeah, we've been allowing crossing to occur for, 10,000 years, and we don't know what genetic changes are happening, and people have been dying young for that whole time, right? They've been living a long time during this time of pesticides and genetic engineering. So I'm playing devil's advocate. I don't really believe that pesticides are good things, but I'm just trying to help you understand that that's the way I see it.
There might not be more cancers now, but now we know about it now because there's more ability where it wasn't that way 60 years ago.
Right. Well, there is more cancer because many times it's a disease of aging. And as we survive, to a certain age, then cancers are more likely to occur. So, but if you want to see, now the National Academy of Sciences report includes an evaluation of the incidence over 20 years, or this time of genetic engineering of crops. 20 years in the United States versus United Kingdom. Or it might have been just focused on England, but either way. Because here we have genetically engineered crops, there they don't. And you can look at the graphs yourself. I was surprised there were I don't remember any meaningful differences between the two countries in all the chronic diseases he looked at. And just by chance, I would expect you to get some. So there is a mountain of evidence, there is a mountain of study on this issue. Hundreds of papers on safety, hundreds of papers on environmental impacts, hundreds of papers on composition, nutritional composition, lots and lots. It's just a huge amount. So, we okay on that for now? Okay. And again, I'm trying to torque your thinking a little on this topic and help you understand that it is bigger than whatever you've probably heard. Bigger in the sense of risks and bigger in the sense of benefits. So I'm going to show you an example of how genetic engineering actually makes foods safer. Okay, so this is a common ear rot in Kentucky and throughout the world. Clint, I know you've seen Fusarium ear rot many, many times. You've dealt with fumonisin. Those are natural toxins and cancer promoters, I should say. Cancer promoters as well as toxins. Bad actors, we don't want them in our food. One reason why is they are a risk factor for certain birth defects. That's not speculative, that's well known. Aflatoxins, you may have never heard of them, but how many times in your career have you seen aflatoxins? How many years in the time you've been here?
In 15 years, probably, it's been an issue 5.
5, okay. Well, that's certainly something that gets your attention. The Down south, it's even more of a problem. And aflatoxins, why do we care? They're natural toxins, all right? This is not anything we've done. But they're extremely potent liver carcinogens. So we don't want these in our food. And in fact, our food system, you know, the big name brand, you know, corn chip makers and masa makers and all that, they're checking for these toxins. And, you know, when they get corn from, you know, Western Kentucky, they're checking for toxins, thankfully, so we can not think about it. But, you know, there are people that have to think about how do we keep these, you know, at low levels. And so this is conventional corn, non-GMO. That is the same corn with a single gene from a soil bacterium that makes a protein that is toxic to certain caterpillars. So the plant makes a protein that is, you would have to eat 2 million, we've done the calculations in my class, you'd have to eat 2 million pounds of corn, of this corn, to get a toxic dose of that protein. It doesn't affect mammals, but it affects certain caterpillars, and so they don't feed on the engineered corn the way they have here, and the mold, the wounds are then allow the fungi to get in. So if I have to choose, I want that corn that's engineered, not that one that has fumonisin on it. So it's actually safer. And here's a study just published a few weeks ago that took our breath away throughout the world of science. This is for aflatoxin. These three are the non-engineered, the GMO, non-GMO corn, and that's a lot of aflatoxin. I don't want that corn. These are all engineered. And so you know, it's really a personal choice. You can have non-GMO, and this is a big problem in Africa and South America and Central America. This is aflatoxin contamination, fumonisin contamination are big deals. You can read all about it. So this, man, I'm telling you, people all over the world, scientists all over the world are saying, wow, I hope that holds up in the field. You know, we'll see if that holds up in the field. That'd be great. And, okay, so I've already made this point. It's a problem in the developing nations. One more on food safety. This is on the marketplace. I don't think it's hit Kentucky. The innate potato. Have you seen the innate potato yet? Okay. It is engineered. So the acrylamide is a natural probable human carcinogen. It's a natural probable human carcinogen in your French fries. And now we probably shouldn't eat too many French fries anyway, but if you do, I mean, I would rather have the one with less acrylamide. And they've engineered a plant using just potato genetics, cis genes, to achieve that. So you'll see that on the marketplace someday.
Are we going to know that's what it is?
Well, it won't be labeled non-GMO. They probably will proudly label it innate potatoes. I don't know how they're marketing it, but this was not Monsanto's trait. This is Simplot's seed company. And their thinking is that, you know, there's the non-GMO, well, the GMO stigma, but there's also a number of benefits, less pesticides, et cetera. And they're betting that the consumer will choose the benefits that are there. So we'll see. Okay, I want to switch gears and talk about, I just checked the time, talk about pesticides. Nobody likes pesticides, right? Nobody likes pesticides. I work with them, but I don't like them. And I try to do work to reduce their use, but I haven't been very successful. So I think if we want to reduce pesticide use, we have to give farmers alternatives for disease and pest control in particular. Same thing. This is a smallholder farmer from Central America. He's not even protected. So can we give him alternatives in that cabbage to make that cabbage resistant? And this is a sad picture. This little girl is about 10 meters away from that rice field, and that rice field gets treated with pesticides by air. So I'd like to see, us come up with ways that farmers have alternatives where they don't have to spray. And so, quick plant pathology lesson. There are, every disease control practice can fit into 4 categories. Okay, one is chemical. We don't like that, so let's X that out. Cultural practices. The way you grow the crop can influence disease development. It's great when it works. It's great. It's important, but it's not enough. Typically, in most crops, it's not enough. Okay, so what else? Biological control. Use organism A to control infectious organism B. Great when it works. Pretty uncommon in our crops to have biological control work. I know you know this yourself. So, we're glad for it, but it doesn't really help very much most of the time. And then, so that leaves genetic resistance. And by genetic resistance, I mean resistance that's genetic, whether it's genetically engineered or conventional. That's the path right there to reducing the pesticide use in that rice field, in my opinion. I'm going to show you a few examples. I've written a lot about this. Yeah, let me show you. This is, all right, I'll take a minute. So this is a naturally occurring bacterium in the soil that produces these diamond-shaped protein structures. I mentioned this before. Organic growers have been spraying this bacterium on fields for years. And there's the diamond-shaped structure. Okay. And so, and that protein is like you'd have to eat just, like a truckload. It's not going to hurt you, but it is going to hurt certain caterpillars, and they die when they eat it. So what has been done, mostly by Monsanto, but I don't think exclusively, is the gene for that protein has been put into certain crops. And corn being a very good example. And look at the insecticide use since it was put into our corn. So there's no question that we can reduce pesticide use with genetic engineering. This is a no-brainer for a plant pathologist. I'll give you a few examples. Here is that same, it's called BT, by the way. Did I say that? Yeah, BT crops. BT stands for the name of the bacterium. So you can pick out, now this is Bangladesh. Bangladesh is a place where eggplant is culturally very important. It's a very important dish for them. So they have to spray like 70, 80 times or more with insecticide. Can you imagine 70 or 80 times?
They'd blow a budget pretty quick.
Yeah, well, I don't know what they do, but they spend a lot of money. I mean, these are farmers without many resources, but that's what they feel they have to do. And they love this technology. It's been released just in the past few years, but it's sweeping Bangladesh because they don't want to spray. Now, they still have to spray sometimes, but their usage has gone down considerably. Here, let me show you a few examples. Rice blast, the most important disease of the most important cereal crop in the world, and he's spraying, and he's not protected. So wouldn't it be nice if we could create a resistant plant? Well, here is an example from genetic engineering. The top 2 are rice leaves from a control plant, and these 3 are rice leaves from an engineered plant. And you might say, well, what did they do to the plant? All they did was they put in a corn promoter. So a promoter is like a light switch. It turns on the gene or turns it off. And so they took a promoter out of corn, that's all it does. So the rice is actually defending itself naturally. It just has its own genetics turned on, its own defenses turned on. This is a case from Florida, lots and lots of bacterial spot, and there's the same genetics with a single gene from pepper. And what does that pepper gene do? It's an R gene. It's a receptor molecule. So that pepper gene, the protein that it produces, just recognizes the bacteria and triggers the tomato to defend itself. So again, the defenses in these cases are actually natural defenses. And I like them better personally. I like that better, much better than what they're doing now, which is spraying copper pesticides. It's not good for the land. It's not good for the workers. Okay. So by now you've probably gotten an idea that I am making sure that benefits get briefly but adequately represented. So now I want to switch to talking about some of the risks and concerns. We've already covered a couple. The food safety issue is really, as far as I can tell, not really a significant risk. And glyphosate tolerance, we talked about that. There are some issues there. I'm going to introduce a few additional issues. So remember, a transgene is a gene that came from outside the breeding pool. And so clearly that can move in pollen or maybe in seed. Okay? Does that represent a risk? I mean, maybe. In some level, it probably does. Now, not all genetic engineering is using transgenes. Remember, there are cis genes from within the breeding pool, and there are these. Remember I showed you the targeted where they knocked out one letter out of a wheat gene? That's not a cis gene, that's not a transgene, that's a targeted mutation. So those kinds of things are not even on the discussion right now. We're just talking about transgenes from outside the breeding pool. A corn gene and a pepper gene in tomato, for example, or corn gene in rice. So there are two broad areas where transgene flow might be a problem: socioeconomic impacts and ecological impacts. The socioeconomic impacts I want to mention first, because I actually think they're maybe the more important ones. This case really disturbs me. I really think it's rather unfortunate. So creeping bentgrass was engineered by the Scott's company, I think in cooperation with Monsanto, to be resistant to glyphosate, so Roundup-ready creeping bentgrass. They obviously thought they had a market for it. They started to produce it experimentally in Central Oregon. Now, creeping bentgrass has got lots of pollen, little tiny seeds. What's the chances that it's going to spread? I mean, pretty good. It did. So that gene, Roundup-ready gene, has landed into weedy populations of creeping bentgrass far from where they released it. Why is that a problem? Because Oregon is the principal grass seed producer for, as far as I know, the world. And they've got markets all over the world: Japan, Europe, and Europeans want GMO-free seed. So there are some concerns about some of the growers there, many of the growers, that this is going to spread into the main production areas for seed. So hopefully not, but it is a preoccupation. Transgene flow can affect organic farmers or non-GMO farmers as well. If they're trying to grow GMO-free corn to sell, and they get pollen blown in from a neighbor, that could affect their certification. So I don't really have easy answers for these. There are things we can do, but we're not trying to do them yet as a society to try to reduce these issues. And here's a cultural issue. These are heritage varieties of corn in Central America, and they don't want transgenes in their corn, in their heritage corn. It doesn't matter whether it's safe. It doesn't matter if there's no ecological impact. They don't want transgenes in their corn. And I would have to take their side on this. So let's talk about ecological impacts, and there are two perspectives here, or two aspects of it. One is the global ruin hypothesis, and what that means, I mean, it's pretty obvious what it sounds like. The idea is that genetic engineering will cause global ruin. As far as I can tell, because they're proposing that genes will run rampant through the natural ecosystems. If it's a valid concern, then I'm concerned. I've taken this very seriously, and I've decided that their idea does not have merit. It's not been published in a peer-reviewed journal, even though they've been talking about it for several years, and I think I know why. I see two problems. One is when you create a transgene, when you transfer a gene from organism A to organism B, you don't create a jumping gene. Jumping genes exist. They're natural. They already exist out in the world. But when we do genetic engineering, we don't accidentally create a jumping gene. We're just, remember, we're moving a sentence from here to here. So you take a sentence that does something you want. You don't engineer a jumping gene. All right, that's one. Maybe even more importantly, they have not accounted for the reality of biological barriers to gene flow in this world. So what do I mean? So genes don't flow freely between plants. There can be some cross-hybridization from species to species out in the world, but by and large, genes don't flow willy-nilly. And the proof is right out that window. Because if you look out there and walk around, you're going to find maybe 100 plant species, not one. If there were free-flowing genes, you would find one species all around. And when you go home, you'll find another 50. So biological barriers to gene flow exist and allow species to exist on Earth. And I pointed this out to the principal protagonist of this directly. And you know what his response was? He insulted me directly. And yeah, and that's really not a very powerful scientific argument. So I've been waiting for a response, and that was it.
What he said?
I don't exactly remember. I do remember responding. I pointed out that that was unprofessional, and I was going to block him. Boom, and I did. So, yeah, he's actually, this individual is known. I mean, he's kind of funny. He's known for bombastic responses when he's painted into a corner. He's very intelligent, but not very reflective. Either way, reflective or not, if he's got a good idea, then we have to take it seriously. They do not have a good idea. It is not. They've got to explain. They've got to get past. They used a lot of math, but they got to get past the biology. They got to explain to biologists why we should expect global ruin. Now, I do expect this, that transgenes could lead to altered species composition in nature. So on the farm, you know, we grow a crop, but outside the farm, there are plants that may comprise an ecosystem of some sort. And, you know, transgene flow could occur into a wild relative of that. And, you know, it's always a possibility. So what are the possible outcomes? So one is the wild relative that receives the genes may do poorly. And so if it does, those genes disappear. The plant receiving the pollen may be neutral, so the transgene will occur, but it won't have an ecological impact. Or the recipient plant may do better, in which case then you would expect a change over time in the species composition and interactions that occur. So it is possible. I do think we should monitor for this and evaluate the risks. It's a reasonable idea if you said, no, I don't want to take the chance on transgenes. I think it's totally reasonable. I see some value in transgenes, but I think it's defensible to say, no, I don't want to take a chance. I don't think we're going to cause rapid changes, but we could cause some changes. And finally, just to put a point on this point, there are many sources of alien genes. Transgenesis is only one. You know, breeders bring in crops from all over the world. We buy foods from all over the world and discard some of the food, and it may sprout and produce flowers. So, you know, there's a lot of sources of alien genes. I don't know, just something to think about. Okay, a few more, I'll flash by, and a few more things that people consider concerns. There's been a lot of consolidation in the seed industry, particularly by Monsanto, but others as well. And that's been driven partly by genetic engineering, not entirely by genetic engineering. Some people are concerned about that. Some people are concerned about patents and their impacts, especially patented seed, especially how it might impact small farmers in developing countries. And yeah, it's a good point. Some genetically engineered traits are actually going to be given away. So I'll show you some of those. Here's some examples. There's very sick cassava roots. It's not going to produce very many if it's sick like that from viruses, and those that have the virus will be, have those brown areas. And this is a real serious problem that no one has resolved yet. This African farmer or scientist has an African solution, and it's a genetically engineered solution, and it's going to be given away freely. There's no patents on this.
Wow.
Yeah, the developing countries typically are developing traits that are going to be low priced or distributed freely. We go skip that. This is another African study that showed 3 weeks without water in the greenhouse. That's like baked. And this one is the non-engineered. There's the engineered. And drought is a serious concern in parts of Africa. This is the non-engineered corn with hardly any kernels. There's the engineered corn. And I don't know if that'll be distributed freely, but I'm pretty suspicious it will. Bacterial wilt of banana. So we've cut open the big stem and see all that goo of bacterial, billions of bacterial cells coming out of that. To a plant pathologist, it's like beautiful, Yeah, we're sick. No, we're interested is really the case. So that kind of bacterial ooze inside the plant causes wilting, and it also causes the bacterium gets into the fruits and rots the fruits. So this is a serious problem, again, without any genetic resistance available. And there's some differences. And African scientists, once again, are heavily involved in this work. They might collaborate with people in the United States or Europe, but it's very much Africans trying to solve their own problems. And that is, this is the last picture. This is to show you that CRISPR has made genetic engineering rather straightforward for people with the skills. And he's actually doing this in his kitchen. Yeah, and he's actually trying to engineer out genetic defects in dogs. So he's got a good, he's got a good heart. He loves dogs. But I looked at this picture and I didn't see dogs. I said bioterrorism. Not that he's doing it, but that's to me, if I have to pick an issue out of this complex of things we talked about, that's the one that worries me, bioterrorism.
Oh, take that and then go with it.
Yeah, engineer a microbe to do something dastardly. Now he's going to not be doing it with an open, breathing in the, whatever's he was working with. But
now,
yeah.
Is there a reason why you have that over there on the table?
Oh yeah. So should we talk about this? Do you want to talk about it? You had the idea. So.
This one is labeled non-GMO, and then the other 5 varieties beside it do not have this non-GMO label. Well, those other varieties of popcorn are also non-GMO. And so this is very misleading to the consumer. They think, oh, I have to buy this one because it's non-GMO. Well, there's not any GMO popcorn on the market. There's not any GMO peanut butter on the market, but these are clearly labeled non-GMO. And so the point that we want to make with this is marketing. This is simply marketing. It is these companies wanting to put a non-GMO label on here. Maybe they can charge a little bit more. Maybe they, again, are feeding into consumers' fears and misconceptions.
So their volume's going to go up because you're just going to
automatically choose that. Yes. And so you all can look at these, it's just, there's some bread, even the oranges. There are no GMO oranges. This package says non-GMO. That's very misleading to consumers. And so that is our main concern is if you choose that you want to stay away from genetic engineering, we absolutely respect that. But we want you to make that choice from an educated standpoint. Okay? We want you to be educated about the food that you're purchasing and consuming. Do you have any questions about this?